So, continuing with the theme of brain injury, it's my pleasure to welcome Gemma Sullivan. Gemma trained in neonatology mainly in Scotland and also in Melbourne in Australia. And she was a clinical research fellow in James Boardman's group at the Clinical Centre for Brain Sciences at the University of Edinburgh. Gemma completed her PhD on the role of inflammation in preterm birth injury, and I'm pleased to say has recently been appointed as a senior lecturer at the University of Edinburgh, and she is going to give us an overview of her work, and her talk is entitled The Role of Perinatal Inflammation in Preterm Brain Injury, an integrated study of immunology, neuroimaging and stem cell modelling. Gemma, welcome and congratulations.
Thank you very much. It's a pleasure to be here to present this work, and I'll talk through the role of perinatal inflammation in preterm brain injury using an integrated approach. So we know that preterm birth is increasing worldwide, and it has an estimated global preterm birth rate of 11%, which equates to approximately 15 million live births each year. And whilst advances in perinatal care have resulted in improved survival rates for babies born preterm, the rate of disability in survivors has not shown the same degree of improvement, and that's largely due to static rates of brain injury. Complications of preterm birth extend across the life course, and we have limited therapeutic options to reduce this risk of impairment. And we can look at brain imaging to provide some insights into the underlying tissue microstructure behind these phenotypes. And here you can see a fetal brain atlas from 22 weeks through to term. And this demonstrates that the third trimester of pregnancy is a period of significant brain growth and development, particularly with regards to cortical size and folding. And this can provide some insights into the underlying biology. Preterm infants are delivered during a critical period of central nervous system vulnerability due to multiple key processes that occur during this window, including neurogenesis and migration, dendritic arborization, synaptic formation and network formation, and the morphogenesis of glia, and of course white matter development. During this critical time, our preterm infants can be exposed to multiple episodes of hypoxia ischemia or infection inflammation, which can occur in utero and postnatally due to the complications of prematurity. This leads to multiple injurious processes, including oxidative stress, excitotoxicity, and activation of the systemic immune response. And this leads to blood-brain barrier dysfunction, influx of peripheral immune cells to the central nervous system, activation of astrocytes and microglia, oligodendrocyte dysmaturation, which leads to myelination failure and neuronal injury. And so this results in a complex amalgam of brain dysmaturation, which includes reduced regional and total brain volume, white matter and grey matter injury, altered microstructure, hemorrhage, and reduced functional and structural connectivity. So one example of perinatal inflammation is exposure to histologic chorioamnionitis, which is closely associated with preterm birth. Microorganisms during pregnancy can ascend from the lower vaginal tract to cross the cervical barrier and invade the decidua and membranes. In some cases, this can extend to the chorionic plate and the umbilical cord, and that is referred to as a fetal inflammatory response. And when histologic chorioamnionitis involves this fetal inflammatory response, this is associated with not only neonatal mortality, but also morbidity, including lung disease, intraventricular hemorrhage, and NEC. We also know that chorioamnionitis is an independent risk factor for white matter injury and also neurodevelopmental impairment in childhood. And so this suggests that organ injury is mediated by the systemic inflammatory response, but the key mediators driving this injury are not well understood. And so that's been the focus of my research so far, to try to identify specific mediators associated with brain dysmaturation after preterm birth. And to do this, I studied 4 main hypotheses. One, that preterm infants have a distinct immune profile at birth when compared to term-born controls. 2, that exposure to chorioamnionitis alters this immune profile, both in cord blood and in the postnatal period. 3, that immune mediators associated with chorioamnionitis also predict white matter dismaturation in preterm infants. And finally, that those immune mediators modulate cortical neurogenesis. And so to do that, I recruited infants to the Their World Edinburgh Birth Cohort study and obtained cord blood and postnatal blood from preterm infants and term-born controls, assessed placenta for histopathology, and identified mediators associated with the fetal inflammatory response. I then looked at the association between these mediators and white matter microstructure, assessed using diffusion tensor imaging at term equivalent age, and then moved on using the immune mediators highlighted from the human studies to look at the impact of those cytokine exposure on developing iPSC-derived cortical neurons, looking at markers of maturation and also neurite outgrowth. So first of all, to assess the umbilical cord profile, I looked at preterm infants who were less than 32 weeks and term-born controls, and they had cord blood dried blood spot obtained for a customised immunoassay of 24 immune-associated proteins, which included interleukins, chemokines, complement, and the neurotrophin BDNF. And this was performed in collaboration with Staten Serum Institute in Copenhagen. And what we found was that 9 immune mediators were differentially expressed in preterm infants compared to term-born controls. In red, pro-inflammatory markers IL-6, MCP1 and CRP were elevated in preterm infants, whilst in black you can see those that were down-regulated in preterm infants compared to term-born controls, suggesting a degree of developmental regulation. And when we performed a principal component analysis, we found that the first 2 components explained the majority of the variance in the cord blood profile. And on projection of the individual inflammatory profiles onto those first 2 components, we can see quite a different pattern for term and preterm infants. And this cord blood immune profile predicted gestation age category with an accuracy of 86%. So moving on to hypothesis 2, looking at the exposure of chorioamnionitis and how this alters the cord blood profile and also the postnatal day 5 blood profile in preterm infants. Placenta histopathology was performed and inflammation was identified using a standardized system whereby maternal inflammatory response was defined as chorioamnionitis or intervillositis, whilst fetal inflammatory response was defined as vasculitis of the chorionic plate or phenocitis affecting any vessel of the cord. And so what we found is that 7 immune mediators were altered in infants who were exposed to chorioamnionitis compared to those who were not exposed, and using regression models to predict this exposure, found that IL-8 was the strongest predictor, and ROC curve analysis showed an area under the curve of 0.917. Moving on to the day 5 profile, we found that although in the umbilical cord the immune mediators were really representative of the innate immune response, on postnatal day 5 there were some interesting observations emerged, including complement activation and neurotrophic capability with BDNF. And so these findings also suggest that there could be a window of opportunity for therapeutic intervention in the days after preterm birth to reduce the risk of complications that we see in association with a placental fetal inflammatory response. And so with that in mind, we then applied a similar approach to assess these immune mediators and their association with white matter dismaturation at term equivalent age. So our preterm infants who had day 5 blood spot obtained also had an MRI scan performed on a 3 Tesla scanner at term equivalent age in natural sleep to assess white matter microstructure using diffusion tensor imaging and neurite orientation, dispersion and density imaging, or NODI. And so I'll just spend a few moments going through some of our MRI tools before we move on to the results. So diffusion tensor imaging can be used to assess white matter microstructure by calculating the degree and direction of water molecule diffusion within individual voxels of the brain. So water molecules move randomly in the CSF in an unrestricted environment, whereas when diffusion is hindered by cellular membranes or myelin in the white matter, water diffuses along the direction of axons. And so we're able to use several diffusion tensor imaging parameters to make inferences about the underlying white matter microstructure. Recent advances in neonatal imaging protocols include increased angular resolution, so we can also apply more complex biophysical models such as the NODI model, which looks at intracellular, extracellular and CSF fractions to give us a neurite density index and an orientation dispersion index. And these values give us insights into underlying structure, including axonal packing and density, myelination, water content, and also geometric organization of neurites. After birth, maturation is characterized by a reduced water content, by increased myelination and increasing complexity, and these measures have all been used to characterize the changes that we see in association with preterm birth. In recent times, these imaging markers have actually been optimized to generate a global measure of white matter microstructure known as peak width skeletonized mean diffusivity, or PSMD. And this is generated using the white matter skeleton, which you can see in yellow, the major information highways of the brain, and a histogram analysis, which is calculated as the difference between the 95th and the 5th percentile. PSMD has been used previously to assess white matter disease burden in cases of multiple sclerosis or small vessel disease, and it's a very good predictor of cognitive outcomes in adults. So this PSMD model has been extended to include the NODI parameters by Manuel Blesa in our lab and optimized for neonatal application. And when we used PSMD and PSNDI, looking at the Edinburgh birth cohort participants, we found that those measures were able to discern between preterm infants, shown in blue, and term-born controls, thereby supporting their use as a generalized biomarker of disconnectivity, which we know is a hallmark of preterm brain injury. So moving on to the results, we found that principal component analysis in the first 5 components actually accounted for around 70% of the variance in the day 5 blood profile, and the immune mediators that were contributing to the variance are highlighted in purple. We used these markers to predict PSMD and PSNDI at term equivalent age, and we found that IL-8 was associated with peak width skeletonized neurite density index, PSNDI. We accounted for gestation age at birth and postmenstrual age at scan, and the model explained 48% of the variance in PSNDI at term equivalent age. None of the remaining 13 analytes showed any association, and there were no associations with PSMD. And so just as a summary recap of the human studies so far, preterm infants indeed have a distinct inflammatory profile at birth. Chorioamnionitis is associated with cord blood IL-8, and placental fetal inflammatory response alters the immune profile in cord blood, but also on day 5. And the day 5 IL-8 predicts white matter dysmaturation in preterm infants. And so this led to the hypothesis, could IL-8 have cell-specific effects to modulate neurogenesis? And so I went on to test this hypothesis using an iPSC-derived model of cortical neuron development. So we know from preterm imaging studies that preterm birth is associated with reduced cortical volume and altered cortical microstructure compared to term-born infants. And we know that those phenotypes persist into childhood and adolescence and are associated with neurodevelopmental outcomes. But the underlying neurobiology behind these cortical signatures are not well understood. We do know that all neurons and glia of the central nervous system are generated from neuroepithelial cells, which you can see in purple. And initially, these undergo symmetrical division to increase the precursor pool. During neurogenesis, after closure of the neural plate, these cells transition into radial glia, which have the potential to become neurons or glial cells. But during neurogenesis, which occurs first, these cells transition into intermediate progenitor cells in green. And again, these can undergo symmetrical division to increase the precursor pool further, or they can terminally differentiate to produce a young neuron. So these processes are tightly regulated, and chemokines and cytokines may perturb these processes, and so that led to the hypothesis that immune mediators associated with the fetal inflammatory response may contribute to this phenotype. And so to assess that, we first generated induced pluripotent stem cells, which can be reprogrammed from adult skin cells using 4 key reprogramming genes to reinstate their pluripotent stem cell state, similar to an embryonic stem cell, which can then be differentiated into any cell type in the body with the right ingredients. And so we use these iPSCs to generate neuroepithelial cells, and then to condition them to continue down the pathway of radial glia, intermediate progenitor, to create neurons. And this process takes around 2 weeks for very immature neurons, which then become physiologically active by 28 days. Using immunohistochemistry techniques, we can label these individual cells so that you can identify them within a culture. So neuroepithelial cells stain positive for nestin, astrocytes for GFAP, and neurons for beta-3 tubulin. And so after plate down of these neuroepithelial cells, or NPCs, on day 0, I selected cytokines that were involved in the fetal inflammatory response, C5a, IL-6 and IL-8, and exposed developing neurons to a 7-day exposure from day 14 to day 21. You can see representative images of the cell culture system with nestin-positive NPCs in magenta and beta-3 tubulin positive neurons in green, and GFAP positive astrocytes in orange. And so the outcome measures of this study were cell populations of each of those, and also neurite length, given the findings from the imaging study. The cells were cultured in 24 well plates on glass cover slips, and data was generated from a mean number of 3 derivations for each condition. And what we found was that IL-8 was associated with an increased progenitor population, shown in graph B, which represents the percentage of nestin-positive cells in the culture, but we didn't see any change in beta-3 tubulin or in GFAP. And secondly, we also found that IL-8 exposure was associated with reduced neurite length. And whilst this phenotype may represent neuronal poor health, dysmaturation, or a functional deficit, given the importance of neurite outgrowth for synaptic formation, for axonal guidance, it's very important to try and identify the functional outputs of this phenotype, especially given the association we've seen with IL-8 and peak width skeletonized neurite density index. So in summary, we've shown that the cord blood immune profile can predict gestational age category, and preterm infants have a distinct inflammatory phenotype. That exposure to chorioamnionitis affects the immune profile at birth and on day 5, which suggests that there could be a window of opportunity for therapeutic intervention to reduce the risk of comorbidities associated with the fetal immune response. Day 5 IL-8 predicts PSNDI in preterm infants at term equivalent age, and IL-8 modulates cell diversity and neurite outgrowth in this human iPSC-derived platform. And so what do we know so far about IL-8 and the developing brain? We know that IL-8 is a key mediator of the innate immune response, where its main role is in neutrophil chemotaxis. But IL-8 acts via G protein receptors CXCR1 and 2, which are widely expressed throughout the central nervous system and have been shown to be involved in critical neurodevelopmental processes, including cell migration, axonal growth, and synaptic formation. We also know that elevated neonatal IL-8 is associated with a number of perinatal brain injurious processes, and neonatal IL-8 has been associated with cerebral palsy following preterm birth, with altered cerebral metabolism and neurodevelopmental outcome following hypoxic ischemic encephalopathy in term infants, and also with the diagnosis of autism spectrum disorder in term infants. Additional studies looking at the maternal immune activation hypothesis have also shown elevated maternal IL-8 during pregnancy is associated with an increased risk of schizophrenia in offspring. So whilst there are several potential mechanisms whereby IL-8 could have cell-specific effects and perturb healthy brain development, further studies looking at causation are warranted, especially given the role of neuronal and glial crosstalk in the context of immune activation. So the 2-dimensional model that I described to you looking at cortical neurogenesis has generated some important insights, but 3D brain organoids really recapitulate fetal brain development, and containing both neuroectoderm and microglia would allow us to look at the real-life activation of microglia and how this can perturb cortical layering and migration. And what you can see on the left here is a work in progress. This is a mini brain organoid containing cortical neurons stained in green and microglia precursors stained in magenta. And so this is early work, but a model that we are hoping to utilised to explore this role of inflammation in preterm brain injury further. And then finally, next steps will involve longitudinal assessment of infants in the cohort with developmental assessment and serial MRI, so that we can, first of all, identify whether early life immune activation affects brain health across the life course, but also to look at the predictive ability of the peak-width skeletonized water diffusion parameters in predicting outcomes after preterm birth. So I'd like to thank both of the labs that I work in, my collaborators, colleagues, supervisors, and of course the babies and families of the birth cohort study and our funders, Their World. And I'd be delighted to answer any questions.
Thank you, Gemma. Amazing work. I think we've been really fortunate to hear about so much important work going on in terms of brain injury, both in terms of in term babies and in preterm babies. So this is open for questions. Yes, question.
Thank you very much. That's just fantastic. I'm Sarah Bates. I work in England. I've got a particular interest in preterm optimisation and trying to reduce the rate of preterm brain injury. And I wonder whether you could speculate on this early therapeutic window and the impact of interventions like deferred cord clamping, potentially increasing the amount of stem cells, and exposure to early maternal breast milk, and the potential impact that those interventions might have in modulating this immune response.
Absolutely, thank you
Thank
for your
you.
question.
Sorry, if I just great question. Thank you. So the question was, how can early interventions such as delayed cord clamping and breast milk affect the processes that you've just described?
We know that optimal cord clamping is very important for stabilisation and would support that. I can't comment on the role of stem cells in brain maturation in that regard, but it's certainly an area of research that we've heard from Linda de Vries yesterday that is being progressed in the context of brain injury due to stroke, for example. And so it'll be interesting to see where that leads, and it may well be a therapy that we consider for all infants if that is successful, but it's too early to say from this data. The second question about breastfeeding is a really important one. Work in our lab has already shown some effects of early breast milk exposure on brain development in preterm infants, and Linda also presented some of those data yesterday. We found that infants who are exposed to greater volumes of breast milk in the neonatal stay had better fractional anisotropy weighted connectomes at term equivalent age. So that really supports that very early use of breast milk prior to discharge home for healthy brain development. So that's certainly very key. And we're hoping to extend that to look at markers of myelination, and so we may be back to present that soon.
Thank you. Ela.
Great work, Gemma. Nicely presented. I'm just interested to hear your views on the impact of sex in the immune profile of the immune.
Sure, thank you.
Sorry. So the question was, what is the impact of sex in the immune profile?
So that's an excellent question and an area that I'm very interested to pursue. So I think we do know that early immune responses differ between males and females, and that's been interrogated using animal models. And we do know that male infants appear to be at a developmental risk due to brain injury in the neonatal period, and that in animal models, looking at microglia responses specifically, that they are certainly different between sexes, and when those microglia are transplanted in the opposite sex, they maintain those individual phenotypes. And so it's an area that is really key for us to explore further, especially given the results of the TIP trial, which showed that indomethacin was in fact efficacious in males rather than female infants in prevention of intraventricular hemorrhage. And that just highlights that there may be differences in immune responses, whether that be peripheral or central with microglia. And it may be in the future that we tailor our treatments depending on sex. And so it's an area that we need to explore further. And that is something that I'd like to take further in the next few years. So.
Thank you. Charles Raw.
Charles, principal. beautiful talk and great work. Thank you very much for presenting so beautifully. We're all aware of interventions we do to preterm infants, which some are modifiable, like the timing of cord clamping, others maybe more or less so, like intubation and ventilation. There's a clear correlation between ventilation and the occurrence of brain injury. What we do is we offer immune modulators late in life, corticosteroids. Some now use low-dose hydrocortisone early for prevention of pulmonary disease. Is there any indication to modulators of the immune response early in life from your work or work of others that indicates it's also brain protective? Sorry.
Thank you. I'll try and summarise that. So the question relates to the early interventions that we undertake, particularly intubation, which can lead to pulmonary injury, and whether early intervention with modulators such as steroids could have an influence on the outcomes. Is that about right?
Mm-hmm. Thank you. It's tempting to move on from the data to support this window of opportunity. I think it's too early to say what that immunomodulator may be. I think we've highlighted with the cell work that there may be cell-specific effects, but certainly need more work to look at individual brain cells in development to see those impacts. So it may be that earlier exposure to steroids could be protective, but we certainly don't know that from these data. And we have to be cautious given what we've seen in previous years using higher dose steroids in early life on outcomes. So although it's early to say that would be the future goal, and some of these models are quite scalable, and so if we did have potential markers that could be identified, we could test them in those systems first before we're moving on to other animal models and human studies.
Chris.
Gemma, I'd just reiterate, fantastic, really wonderful to see all of those different kind of modalities of work. Related a little bit to Charles's point, you measured in your cohort of babies exposed to chorioamnionitis, you measured a whole host of different measures, but one of them was CRP, I noticed in there, and it was flagged in several of your slides as being associated with some of your measures, your outcome measures. Is there going to be a role for CRP being useful, an early measure of CRP? Because this is obviously something that we all do measure, and I think there's a lot of scepticism about how helpful it can be. Are you suggesting maybe that there is a role for this?
So this is CRP raises its head once again. So Chris asked the question: what is the role of CRP? And that was something that you referred to on a number of occasions. And does it have a role in this process?
Okay, thank you for your question. I think that CRP came up in the initial slide where I looked at the differences between preterm infants and term bone controls, and so that was elevated in preterm infants. But of course they have a much higher incidence of exposure to intrauterine inflammation, and so when we looked at that properly and I did the regression models to predict exposure to chorioamnionitis, in fact IL-8 performed better. And other studies have shown that IL-8 can also identify infants who have sepsis or have necrotizing enterocolitis. So I might suggest that IL-8 might be a more promising marker of inflammation in the neonatal period.
Interesting. Don.
Hi, and again, just to reiterate, it's a great journey, great to see that going through all of that work. That's nice to hear that you're going on in a great centre and taking that further. In Nottingham, we've been looking at the impact of hydrocortisone and caffeine and things on lung cells, the ability we can stretch the lung cells in the lab. And we know that caffeine interferes with the inflammatory system, and IL-8 in particular, as the systems out there that show effects. Clearly, it's one of the few interventions in neonatology that has shown good trials that we can actually improve neurodevelopmental outcomes. Have you exposed yourselves, for example, to caffeine and seen any differences?
Thank you. Good question. So this relates to the influence of caffeine in neurodevelopment, which is clearly something that is very important.
Absolutely. Thank you for the question. It brings up an area that for future research would be certainly very feasible and scalable. And I have not exposed the cells to caffeine to date, but that is something that we could do for many drugs that we use in neonatology, and also thinking about neuroprotective agents in term brain injury too. So I think it's important, particularly that we have drugs that have off-label effects, as you've described there, looking at IL-8 and inflammation. And so there is an opportunity for us to study drugs in isolation or in combination using the in vitro model. I think that's an opportunity as a community to do that. And so we should talk more about that again.
Any other questions? Yes.
Speech of time. Am I right in your cohort you had babies who were exposed to, fetuses that were exposed to chorioamnionitis and those that weren't? Did you image the ones that worked, and were there differences in the brain microstructure between those exposed to chorioamnionitis and those not? Because clearly those not exposed to chorioamnionitis also presumably have some degree of brain injury. And how do you control for exposures after birth versus before?
So what is the difference between brain microstructure in those who are exposed to chorioamnionitis and those who haven't been?
Thank you. The question that we assessed using the brain imaging was to pinpoint really what are the mediators associated with chorioamnionitis that are associated with brain dysmaturation. So we used IL-8 as a proxy marker of exposure to chorioamnionitis, given that that was elevated in the cord blood and on day 5. And so that really looks at it as a continuous variable rather than a binary yes or no. And we found that the IL-8 was associated with white matter maturation using the PSNDI. And so I would say that is a proxy marker of exposure to chorioamnionitis in this study. You asked the second question, which was about confounders after the exposure to inflammation. And looking at the cohort, we did look at infection rates, at BB day rates. And for those who were exposed to chorioamnionitis and not, there were more babies who developed BPD in that group, but that was described in the paper, which is in Brain Behaviour and Immunity.
Thank you. Any other questions? No, we don't have any online. So, Gemma, I want to thank you once again for presenting a fantastic body of work, and we look forward to hearing more about your work in the future.
Thank you very much.
Congratulations.
Thank you.